Hi, my name is Jeff Hoffman, and in this lecture I'm going to cover the topics in molecular biology that you need to know for your Step 1 exam. The first topic is DNA. There's a lot of DNA in a cell. In fact, if you stretched all the DNA from a single cell end to end, it would be about a meter long. Since a cell is only a few microns in diameter, it needs a way of packaging all this DNA into a small space. So how does it do this? Well, by coiling the DNA around proteins called histones, which are shown here. There's a few different types of histones, called H2A, H2B, H3, and H4. These aggregate together, and DNA wraps around them twice to form a nucleosome. There's one more type of histone, called H1, which is not in the nucleosome, but along the DNA that links nucleosomes together. So what causes DNA to wrap around histones? This is because the DNA is negatively charged, and histones are rich in the positively charged amino acids, arginine and lysine. The lysines on histones are particularly important because gene transcription is often regulated by covalently adding or removing methyl or acetyl groups on these lysines, which changes their affinity for DNA. Now, the effect of acetylating or methylating amino acids on histones depends on the specific amino acid and the specific histone, and you don't need to know that level of detail for the Step 1. But in general, adding acetyl groups or removing methyl groups allows it to be more accessible to RNA polymerase and other transcriptional machinery. This is known as euchromatin, which you can see here. In the opposite situation, having more methylation or less acetylation results in tighter binding of DNA to histones so that it can't be transcribed, and this is called heterochromatin. So which is transcribed more, heterochromatin or euchromatin? That would be euchromatin, since it's more accessible to the transcriptional machinery. I remember this by thinking that acetyl groups are much larger than methyl groups, and therefore will push DNA away from histones, whereas methyl groups are small and allow it to stay close. That's not actually the mechanism, but it might help you keep these straight. You can also use the mnemonics in the book. Histone methylation mostly makes DNA mute, and histone acetylation makes DNA active. Keep in mind this is referring to the methylation of the histone proteins, not DNA. Cells can also methylate DNA, specifically at sites called CpG islands, which are just places in the genome enriched for C's and G's. Why is it called CpG? The P represents the phosphate which separates one nucleotide from the next. Specifically, the cytosine and adenine nucleotides would be methylated within CpG islands. Okay, so why would a cell methylate CpG islands? This is just another way that cells can repress transcription. If you know you're not going to need to express a gene for a while, or maybe even ever again for this specific cell, methylating a CpG island and its promoter is a good way to make sure you don't transcribe it by mistake. Now let's talk about the nucleotides that make up our DNA. There are two types of nucleotides, the purines, which are adenine and guanine, and the pyrimidines, which are cytosine, thymine, and uracil. Uracil is only found in RNA, and thymine is only found in DNA. You can remember these using the mnemonics pure as gold, since the purines are adenine and guanine, and cut the pi, since the pyrimidines are cytosine, uracil, and thymine, which spells cut. Now fortunately, you don't need to memorize the structures of these for your exam, but there are some features you should be familiar with. First of all, the purines have a double ring structure, whereas the pyrimidines have only a single ring. You can again use the pi mnemonic to remember this, since pyrimidines look more like a pie. The only other structural thing you should know for the Step 1 are that thymine has a methyl group, here, and cytosine can be deaminated to form uracil, which I'll talk more about in a minute. The differences in the locations of ketones and amine groups determine where and how many hydrogen bonds are made when nucleotides pair up in a double helix. Guanine and cytosine pair up by forming 3 hydrogen bonds, whereas adenine and thymine, or adenine and uracil, only form 2. This is important because it's one reason that GC bonds are more stable than AT bonds, which means it requires more energy to separate DNA strands that are rich in G's and C's. This ties into the concept of the melting temperature of DNA, which is the temperature you have to reach to separate the two strands. DNA is double-stranded at lower temperatures, but as you heat it up, you can get the strands to separate. So would GC-rich DNA have a higher or lower melting temperature than AT-rich DNA? It has a higher melting temperature, because again, you need more energy to separate the strands. Can you think of other properties of DNA that might affect its melting temperature? One of the important ones is DNA length. since shorter stretches of DNA are easier to melt apart. This kind of makes sense since there are fewer total bonds, right? Okay, so who cares about how strong DNA bonds are? Well, we'll come back to this in the lab techniques section, since in some types of experiments or diagnostic tests, like PCR, the melting temperature of DNA is very important. Alright, to drive this home, let's do an example. Which of these pieces of double-stranded DNA has a higher melting temperature? I'll give you a second to think about it. In this case, it would be the bottom strand, since it's both longer and has more G's and C's, both of which help the strands stay together and increase the melting temperature. You should also know what components are used to build nucleotides. I'll come back to this in the next slide when we go through nucleotide synthesis in more detail, but for now, know that they're made primarily from amino acids. Purines require aspartate, glycine, and glutamate, and pyrimidines require aspartate and carbamoyl phosphate. It's convenient that the first letters of the amino acids used to make purines are all A's and G's, since purines are guanine and adenine. One last thing I want to cover here is nomenclature. We've already covered the names of the bases, but nucleotides are more than just bases. They also need to have a sugar and a phosphate. What kind of sugar is it? In RNA, it's ribose, and in DNA, it's deoxyribose. And what's the difference between those? Well, in this diagram, you can see that this 2' carbon in the sugar can have either a hydroxyl group, which makes it ribose, or just a hydrogen, which makes it deoxyribose. So when we say a nucleotide, we're talking about the base, the sugar, and one phosphate. If you have just the base and the sugar, it's called a nucleoside. This also affects the names of specific bases. For example, the nucleoside version of adenine is adenosine, and the nucleotide version is adenosine monophosphate. If you add 2 more phosphate groups to adenosine monophosphate, you get adenosine triphosphate, or ATP, which, as you probably know, is the main energy currency in the cell. Next, let's talk about how these purine and pyrimidine bases are synthesized. These pathways are a little complicated, but they're important to know because some of the steps in these pathways are targeted by treatments for cancer, since cancer cells need to produce lots of nucleotides in order to replicate. Also, a lot of bacteria have different versions of some of the enzymes involved, which allows us to target them specifically with drugs. First, let's take a look at the chemical structures of the purine and pyrimidine precursors. We'll start with PRPP, or phosphoribosyl pyrophosphate. By the way, what does pyrophosphate mean? It's just 2 phosphate molecules linked together. Now this is going to be one of many examples in this chapter where you don't need to know the molecular structure of something, but being able to appreciate its general properties will help you understand the bigger concepts. Okay, so how do we make PRPP? We start with the ribose 5-phosphate, and the enzyme PRPP synthetase will take 2 phosphates from ATP and add them on. So the ATP becomes AMP, and the ribose 5-phosphate becomes PRPP. You can already see from looking at this how it might be converted to a nucleotide. Since, as I mentioned in the last slide, a nucleotide is a combination of a base, a ribose, and a phosphate, and we have the ribose and the phosphate here already. Now why do we want this pyrophosphate on the other end? It's actually for the same reason that ATP has a few phosphates on it. This creates a high energy bond right here where we're going to add the base. So now when we do add the base to PRPP, which is going to go right on the same carbon that the pyrophosphate is on, we already have the energy to do it. We'll lose these 2 phosphates when we add the base. Okay, so PRPP is used to make both purines and pyrimidines, but we'll start with purines because they're easier. For purine synthesis, we're going to add a precursor base called hypoxanthine, which looks like the other purine bases, onto our PRPP, which results in a nucleotide called IMP, or inosine monophosphate. From here, modification of functional groups on the base, hypoxanthine, will convert this either to AMP by converting this ketone to an amine group, or GMP by adding an amine group here. Alright, that's it for purines, not too bad, right? Let's move on to the pyrimidines. These also use PRPP and also need another precursor called orotic acid, or orotate, shown here. It looks kind of like a pyrimidine already with that single ring, huh? Orotic acid is made from a molecule called carbamoyl phosphate, which is also involved in the urea cycle, which is covered in one of the later biochem lectures. The only thing I want to mention about that here is that if someone is deficient in the enzyme ornithine transcarbamylase, or OTC, they'll have a buildup of carbamoyl phosphate, and therefore of orotic acid as well. OTC converts carbamoyl phosphate to citrulline, which then enters the urea cycle. Getting back to pyrimidine synthesis, orotic acid is combined with PRPP, with the help of the enzyme orotate phosphoribosyltransferase to produce OMP, or orotidine monophosphate. This is converted to UMP using the enzyme orotidine decarboxylase. I won't go into the details of how this is converted into UDP and CTP. Basically, you're just adding phosphates and making a functional group change. One thing that is important, though, is that so far I've only shown you how to make ribonucleotides, which are used in RNA. So what's the only difference between a ribonucleotide and a deoxyribonucleotide? It's this 2' hydroxyl group, which is only present in ribonucleotides. So you can see here in PRPP, there's a 2' hydroxyl, since this is used directly to make ribonucleotides. But deoxyribonucleotides are created using the enzyme ribonucleotide reductase, which removes that hydroxyl group and replaces it with just a hydrogen. Since thymidine is only found in DNA and not RNA, ribonucleotide reductase is used along the way. This last step, catalyzed by thymidylate synthase, is especially important for a lot of reasons, including nutrition and pharmacology. Thymidylate synthase is how we make thymidine nucleotides. As you can see from these structures of uracil and thymine, the only difference between them is this methyl group. So we have uracil and want to make thymine, so how do we add this methyl group? Well, one of the most common ways to add a methyl group in biochemistry is to use the coenzyme tetrahydrofolate, which in this case donates the methyl group to uracil to create thymidine. After donating this methyl group, tetrahydrofolate becomes dihydrofolate, but the enzyme dihydrofolate reductase restores it back to THF so it can be used again. Since folate is a key part of tetrahydrofolate, people who have a deficiency of folate can have defects in nucleotide synthesis, which in adults can cause anemia, and what's this going to do in a developing fetus? Folate deficiency causes neural tube defects, so make sure patients who are pregnant or might become pregnant are eating enough leafy green vegetables or taking a folate supplement. Okay, so this is all very interesting, but why do you need to know about it to be a doctor? Or how is this going to show up in your step 1? Well, it's important that you know how we can perturb these pathways and enzymes to treat disease. So what general categories of diseases can we treat by targeting nucleotide synthesis? Well, a major one is cancer, since cancer cells divide quickly and therefore require lots of nucleotides for DNA synthesis. But also bacterial infections, since bacteria mostly use the same enzymes that we do, but have slightly different versions allowing us to target them and prevent them from dividing. What other types of cells divide a lot? Cells in the immune system. So targeting nucleotide synthesis can also be used as a very aggressive immunosuppressant to treat inflammatory or autoimmune diseases. Let's go through some examples. There's a few drugs that target dihydrofolate reductase, but what's the difference between these? Well, methotrexate targets the human version, so it's used to treat cancer, whereas trimethoprim specifically targets a bacterial version of dihydrofolate reductase to prevent cell division in bacteria without having much effect on the patient. Pyrimethamine is specific for protozoa. And lastly, 5-fluorouracil inhibits thymidylate synthase, which again is to treat a variety of types of cancer. So here's a question for you. How does taking folate, or folinic acid, affect the drugs that interfere with this cycle? This is something that always confused me when I was studying for the step 1. Folate is often given with 5-fluorouracil because it enhances the binding of the drug to its target enzyme, thymidylate synthase, making it more effective. In the case of methotrexate and trimethoprim, which normally cause folate deficiency, it's used to reduce side effects, especially in tissues that need lots of cell division such as bone marrow and gastrointestinal lining. So if this comes up in your exam, in both cases you do want to give patients folate supplements when you prescribe any of these drugs, but for 5-fluorouracil is to increase potency, and for methotrexate and trimethoprim is to reduce side effects. Hydroxyurea inhibits ribonucleotide reductase. And what's that going to do? Well, ribonucleotide reductase is needed to convert ribonucleotides into deoxyribonucleotides. So again, this prevents DNA synthesis, and this is used in humans to treat cancer. But hydroxyurea also has another really important clinical use, which we'll cover in the Hem/Onc section. Do you remember what it is? It's actually one of the main things used to treat sickle cell anemia. So how does it help these patients? Well, remember, sickle cell anemia is caused by a mutation in adult hemoglobin. But hydroxyurea causes these patients to produce more fetal hemoglobin, which reduces their frequency of sickling cells. So this is used prophylactically and as long-term therapy. You basically start them off in a low dose and increase it until it's about as much as they can tolerate without developing cytopenias. Okay, next we have 6-mercaptopurine, which interferes with purine synthesis. and this is used to treat leukemias, but also it's used as an immunosuppressant to treat arthritis and inflammatory bowel diseases, because remember, cells in the immune system also tend to divide a lot. Leflunomide inhibits the enzyme that converts carbamoyl phosphate into orotic acid, which is dihydroorotate dehydrogenase, and is also used as an anti-inflammatory, and specifically for a certain disease. Do you know which one? This picture is a hint, and the answer is rheumatoid arthritis. The last immunosuppressant here is mycophenolate, and this one is mostly used with organ transplants to prevent rejection. This inhibits IMP dehydrogenase, which converts IMP into GMP. As you can see in the book, there's a second drug which targets the same enzyme, ribavirin. But this is used for a very different purpose. Did the name give it away? This is used to treat viral infections, including influenza and hepatitis C, but also some viral hemorrhagic fevers. Alright, here's our first flash quiz. We'll be doing a lot of these throughout the chapter, and if you've seen other chapters already, you've seen these before. But I'm going to ask you a quick question, and I'll give you a few seconds to answer it. Alright, so here's the question. Methylation of CpG islands has what effect on transcription? So the answer to this one is it represses transcription. Remember, methylation makes DNA mute, and actually methylation of histones generally represses transcription as well by making them heterochromatic instead of euchromatic.